Hello, my name is Valeria Grazu. I'm a tenured scientist in the Institute of Nanoscience and Materials of Aragon from the CSIC and UNISAR, University of Zaragoza. It's a mixed institute of these two institutions. I'm working since long time in biofunctionalization of nanomaterials for biotechnological applications, in particle nanoparticles, and in particle for hot science projects. I'm working a lot with magnetic nanoparticles. And my role in hot science is I'm the coordinator of the project. So I'm in charge of the management, of the scientific management of all the projects, and specifically, technically, my laboratory and the team of scientists that are working in my laboratory, we are in charge of trying to attach enzymes to magnetic nanoparticles in order to activate them using magnetic heating. Our specific role is to try to combine these two different words that are enzymes, that are proteins, with inorganic nanoparticles, and to do this combination, this binding, without losing the properties of each of them, the properties of the enzymes and the properties of the nanoparticles. So this is our specific role within the project. Well, enzymes are biocatalysts. They are the one responsible that make that all the chemical reactions that occurs in living animals or plants, in living organisms, occurs in the adequate speed. So enough. So they are the catalyst of all the reactions that occur in our cells. They are very specific and very selective, and they could work under conditions that are very environmental friendly. So you don't need to use very high temperatures, so they could work and catalyze a reaction, make that a reaction occurs in a fast way. The problem is that the enzymes works well within our cells. But if we want to extract these enzymes and use them in an industrial environment to catalyze chemical reactions that are used to produce chemicals of interest, for example, drugs, or for example, food additives, these enzymes, it's difficult to pair them, to work, they will work together well. Okay, for example, for the production of certain drugs, I need perhaps to put together several enzymes to work in the same vessel of reaction, and they have different needs. For example, one enzyme could work at 70, need to work at 70 degrees to be very fast, and others at 20 degrees or 30 degrees. So it is difficult to work with these enzymes in an industrial environment and to catalyze a chemical reaction of industrial interest. Okay, so the idea of hot science, the general overview, is to try to attach these enzymes of industrial interest to magnetic nanoparticles. And why? Because it is well known that magnetic nanoparticles will produce heat very locally, very near the surface of the particles. So the idea is to try to heat each enzyme to its optimal temperature. So the idea is to try to use magnetic heating to be able that different enzymes that need to work together will work together, each of them at its specific temperature, optimal temperature to work with. So in the middle of this idea, that is hot science is a proof of concept, we need to see if this is feasible. Okay, this way we could use enzymatic cascade in industrial process that today are not possible using traditional way of production of these chemical reactions because they are very inefficient or energy consuming. Okay, so usually if you have to produce from a starting material, you have to obtain a product of interest, for example, a drug or a food additive. Okay, so you will need to combine, usually to obtain these products of interest, you need to combine different enzymes to be able to combine enzymes in what is called a cascade, an enzymatic cascade. The first enzyme is the one that transforms the starting material in a intermediate product that is used for the second enzyme, and this way, at the end of the cascade, you have the final product of real industrial interest. Okay, so if you have enzymes that all work at the same temperature, it's easy. You could heat up the vessel of reaction with heating, okay, with usual heating mechanisms, okay. But the problem is when you have enzymes that need to work to different temperatures. One need to work at a higher temperature than others, okay? So in this way, you need that the vessel of reaction, the global temperature, it's maintained at room temperature, for example, and locally, each enzyme is heat up, okay, at different temperatures. To do this, the idea is to use magnetic nanoparticles, link the enzymes to magnetic nanoparticles. Why? Because we know that when we apply a magnetic field that is alternating, its direction is alternating, we could heat the nanoparticles. We transform these nanoparticles in heaters, in local heaters. The heat is very, very near the surface of the nanoparticle. So we are able to heat up what it is attached to the nanoparticle, but not all the vessel, okay? So this way, each nanoparticle will heat up different enzymes, and each enzyme will be heat up to its the temperature that it's optimal for each of them. So this is the idea of hot science, to try to have these enzymes cascades working in a coordinate way in one vessel of reaction. So we put the starting material, we combine the nanoparticles with the different enzymes that are needed to produce a product, and we apply this alternating magnetic field, and the idea is to be able to obtain the product with high efficiency. Okay. So for the objective of hot science, that is to be able to control or to make that a cascade of different enzymes having different optimal temperatures could work together, we need to activate these nanoparticles to make that they could start to heat locally each enzyme that is attached to them. For this, we couldn't use the usual enzymatic reactors that are already used in the industry. We have to prepare specific bioreactors that could be that have a control, a remote control of the nanoparticles. So how this is going to be done? So these bioreactors could has to have the feature of being able to apply an alternating magnetic field. This alternating magnetic field is what is going to trigger the activation of the nanoparticles. That means that the nanoparticles start to work as heaters of the enzymes that are attached to these nanoparticles. Okay, so to prepare these bioreactors are not existent now in the industry. They have to be designed from zero. Okay, so this is the role of the company Nanoscale Biomagnetics, and the CEO of the company is Nicolas Casinelli. So this is a company that has been working long time ago in the use of magnetic heating for therapy, for cancer treatments. So their role is to adapt the experience that they have in preparing equipments that could apply alternating magnetic fields, but for therapy. Instead, that for therapy, apply this knowledge to produce bioreactors that could apply alternating magnetic field, but for biocatalysis. That is to be able to activate magnetic enzymes attached to magnetic nanoparticles to obtain a specific product of industrial interest for from a substrate. Okay? So they have to. This is their role. So we need not only to obtain these magnetic nanoparticles with the enzymes needed for the biotransformation, but also these bioreactors that could apply an alternating magnetic field. So these are two very big objectives of Hot Science project. We want to show the feasibility of a new technology, a new way of heat enzymes, okay? So in the nature, we have a toolbox of enzymes that could be combined and practically catalyze any chemical reaction that we could imagine. So in theory, this technology, it works well. It could be used for a wide myriad and variety of chemical reactions to produce a wide variety of products of industrial interest. As we don't have, but in Hot Science, we need to select specific biotransformations to show that this is feasible, okay? So we have select two examples or two biotransformations. One is to produce an intermediate of an antitumoral drug, okay? And the other is to produce a food additive that is used as a sugar replacement, okay? A traditional edulcorant, okay? So we have selected these products of industrial interest because the traditional way to produce both of them are very inefficient. So we select specifically very inefficient biotransformations that currently these products of industrial interest couldn't be produced. And the idea is to show that with this new technology of magnetic heating, and we could improve the efficiency of the process, and thus the feasibility of the industrial implementation of this magnetic heated enzymatic cascades for the production.